yes. And
Hi, Hannah. I'm sorry you're sick.
It's like 5 years without charging it, found it 1% still alive.
Still standing.
Yeah, we now have the charger and everything, we still play it.
You'd never get that battery life today.
Okay, so Eli told me he thought the test was pretty easy. What did y'all think?
I did not think it was easy.
Really? I think it's like one of the easiest tests.
It's
I
easier than
think
the 9
it was
test.
Just like, I guess I didn't, like I did quiz it like a lot of times, but I guess I wasn't like, the stuff that wasn't on quiz, I was like,
Okay. Oh, okay.
I don't know.
You thought it was hard.
Not like hard to think of. It was just like some of the stuff I couldn't remember. Cause
Okay.
like I don't think it was on Quizlet, so it was like.
What did you think, Ashley?
I'm a little scared about the paragraphs, but other than that, I feel good. I feel pretty good. Like the paragraph, like
Oh, yeah,
the
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 paragraph.
discussion
one
question.
paragraph where you had to come up with the ecosystem.
It was more like the true or false for me.
Okay, so
Three
if you think
sentences
about that,
per
that was all like kudzu, the rabbits in Australia, the deer. You could have taken any of those ones that we talked about and just kind of described it and given a possible solution, like bringing up wolves on the island. So, or you could have just made one up.
Yeah, I did the kudzu.
You did kudzu, yeah. And
Cause
then,
I remembered it.
okay, okay. Yeah, I didn't need, Essie, this makes you feel better, I didn't need like a ton of detail about it. I just needed to make sure you understood the gist of an ecosystem getting out of balance, and maybe that it got out of balance because it didn't have a natural predator, and some ideas for getting it back in balance. That's what I was looking for. I bet you did better.
I always do better than I think I do.
I bet you did. You were looking like you thought it was not a good test.
Oh, it's fine. I think I'll get a good grade on it, but I didn't have like SC. I mean, sorry, Ruthie. I couldn't remember it.
You couldn't remember some of the
Yeah.
details. What'd you think, Anna?
It was the easiest.
Okay, okay.
It
Well,
definitely was the,
well, and I actually
Split down the
changed
middle.
some questions and made them easier. Then I went back and looked from last time I taught it.
Oh, Monday was like my worst sickness day, so
That probably
it
didn't
was-
help.
yeah. But it was the only thing I had to do for school, and
Uh-huh.
plus some stuff from other class. But I feel like I did well, but I don't know.
Okay, all right, well, and then everybody's turned in their Module 10 test. You were also supposed to turn in your Module 10 study guide and the Appendix B summary. And I understand that summary thing is a new thing to turn in, so you're gonna send that to me today. Did everybody else turn in the summary?
like this kind of summary.
Mm-hmm.
I didn't
Okay.
know that was.
I lost the original thing that you gave us.
Oh, that's
I just
fine.
you just scanned it on.
Oh, yeah, oh, that's fine.
Yeah.
So Anna, yes, I put that, it was on the calendar and on the email to the moms, and I said something about it last week, but if you have it, if you could take a picture of it or find it.
I'm pretty sure I have it in
Okay.
mine.
Okay, perfect. Well, you can show it to me today, or you can send me a picture. But from now on, I think most modules, I am having you turn those in, and the second semester, I make a lot of test questions directly from those. And so,
I have it.
you got it? Okay, perfect. Yay, okay. Thanks,
Are
Anna.
you allowed to check your answers on your?
Yes,
summary?
yes.
I did it. And
I want
was
you
out
to.
in class.
Yes, I brought the book to make sure you guys understood with these module summaries. The answers are in the same book that the study guide answers are. Yes, check them because you're supposed to use them to study for the test, so you need to make sure that you have the right answer. And sometimes the answers to these are, like, there could be several things that would work. But yes, it's definitely good. I want you to check it and make sure.
I did this last one. And I was like.
And actually, so I went ahead and made the copy today because I felt like while some people were working on the microscope and you had time, what we're gonna cover today is 1 through 8. And so after you hear me talking about it, you could actually just sit and work on it, or if you had time this afternoon or in the car on the way home, you could go ahead and fill in the blanks for 1 through 8 and see how much you remember from reading or class. Okay? I love the videos from this section so much, and I was thinking about how great they were, and last night I had a dream that I came in today and y'all all told me that no one watched any of the videos, and I was so mad. So, but the first thing Eli said one of the first things he said when he walked in was that he watched all the videos, and so I know that's not the case, so I'm happy. And then I'm going to add another, I think it's 4-minute video for tomorrow. I meant to bring my computer today because I watched this other video yesterday that's the jellyfish life cycle close up, and it's so cool, and we were gonna watch it in here today, but I forgot my computer, so I'm gonna send that for you to watch tomorrow, but I think you'll think it's really cool. So we're starting on the what kingdom?
Animale. I don't know how to
You
pronounce
can
it.
just say animal.
I said animale.
You can just say animal. So the animal kingdom, and this module is going to be the invertebrates, the animal groups that have no backbone, okay? And then there's going to be 3 modules of vertebrates, and then we have 2 modules of plants. And so this is gonna be our only module on the invertebrates. So really, the rest of the semester is very much like we're gonna study different phylums of animals, or 2 modules we'll be doing plants. And this is kind of our module where we're gonna be doing some dissections. So, what is the name of the phylum that the vertebrates are in? Chordata. Okay, so all the vertebrates are in chordata. Are there more invertebrates or vertebrates in the animal kingdom? Yes, many more invertebrates. Good. Okay, so we start to learn different ways that we can classify animals into groups. And one of them, what the module introduces on pages 330 and 331, is looking at the symmetry and how the animals are symmetrical. And so we see that in figure 11.1, you see the man, the person up there, and there's a line drawn through his body. What kind of symmetry is that? Where it's bilateral symmetry, where it's equal on both sides. Now, Ruby, is the right side exactly like the left side? No, especially when you look at the insides of the body, okay? So it's not exactly alike, but it's very similar, and so overall, it's considered to be symmetrical bilaterally. All right, so then we had, this was a throwback to when we were studying the bacteria, the volvox. And the volvox has a spherical symmetry. So when an organism is shaped like a sphere, it's gonna have a spherical symmetry, so that if you slice it anywhere directly through on that sphere, each side is gonna be, it's never gonna be perfectly symmetrical, but very close. All right, so then next to our volvox, we have our friend the anemone, who is in a mutualistic relationship with the anemone, the clownfish. Okay, and can somebody explain the kind of symmetry that the anemone has? Eli?
It's radial symmetry.
Mm-hmm.
It's where if you looked at it from like a bird's eye view and then cut it in half, it would be the same.
Right.
And
Right.
all the way around, like
Yes.
spherical symmetry.
Yes, so it's kind of like, anemones are shaped kind of like a tube. Things that have radial symmetry are gonna have some kind of a tube-like shape. And so you could cut it any way straight down and you're gonna have symmetry kind of on either side. Does that make sense, Nancy? Okay. All right, and then next to it, we have the flatworm, which is like the person that has the bilateral symmetry. Okay, what about the sponge? What kind of symmetry does it have?
Radial.
Okay, no
Oh,
symmetry,
no.
okay? So sponges are not going to have symmetry. All right, And what is the phylum name for sponges?
Purifera?
Porophora. Okay. So you can think about it like this: sponges have pores or little holes, or you can think about how you use a sponge maybe to wash your face, to clean out your pores, or something like that. But so I think that one's kind of an easier phylum name to remember. And again, sponges have no symmetry. Are they mobile? Do they move around?
No.
No. So does anybody remember the term for something that's not mobile, that stays in a fixed position? Essie, you look like you've got it on the tip of your tongue.
I remember where it is.
Anybody remember?
I don't remember learning, but it
I think we learned that had this vocabulary word.
Not stationary.
I think we had it when we did Protista.
The kelp, is that what?
Yeah, but we had it, yes, so we had this word,
Oh,
this
my
word was
word.
back.
Hold up, we go.
Okay,
It's like,
but it's in this chapter too.
it's when it like reaches out and sticks to.
So sessile.
Okay.
Oh.
Sessile, okay, sessile is,
Sessile.
yes.
Oh.
Sessile is gonna mean that it is stationary. Okay, so, can anybody tell me a little bit about what they learned about the sponge from either the video or reading about it? Anybody want to describe it? Well, I'll start with Nancy and we'll go down. Okay.
Okay, so I was kind of confused, but what stood out to me was like they used real sponges instead of synthetic a while ago. But I was kind of confused because some places are like they use dead sponges.
Well, when you're using a sponge for
Yeah.
cleaning or something, and these are real sponges,
Yeah.
they're dead. Once you are getting them out of the ocean and moving them, they're dead.
Okay.
Yes, so they are not alive anymore, but
But
that's
they are
still a
in
real
nature.
natural sponge. It's a synthetic sponge is like something that's like made in China that,
Yeah, right.
yes,
If you don't cut off the whole sponge, wouldn't it still be alive and wouldn't it grow back?
yes, yes.
So
And
why are people just cutting off part of the sponge?
well, and so one of the I think I must have had a video last time I taught this because it wasn't on the sponge video, but because sponges can do asexual reproduction and they can kind of bud and regenerate. They're not like as amazing as like the hydra, but they can regrow sections. These sponge harvesters, they know how to. They're not taking the whole sponge. They're taking parts so that it's regrowing and then they're going back to certain areas. So they've learned, they understand the science of sponges and they're trying to maximize what they're getting out of the sponge. So they're not just getting them completely. That makes sense. Okay, Ruby?
So how they get their food is that they're constantly pumping water into like their pores or whatever, and then the water goes back out, but the food that comes in with the water stays, and that's how they get.
Yes. So you saw the video with the dyes.
Yeah.
So you
My
can
mom
see
and
it.
I were watching it. It was so
Did your mom like the videos? I bet she.
Yeah, well, she saw the sponges and she used to live in Tampa, Florida, and that's where they like. What's the word for like getting sponge? Well, I guess
Harvest. They
harvesting
harvest the
stuff,
They harvest sponges.
Yeah.
Okay.
Yeah, there a lot.
Uh-huh.
Or like some bay in Tampa, but yeah.
Uh-huh.
Tampa Bay. Well, yeah, but
Someday.
yeah, but she thought it was really interesting and like the dye going in.
Yeah, isn't that cool?
Yeah, it
Yeah.
reminded me of like almost like a chimney in a fire.
Yeah.
That's what he said.
Yeah.
No, I feel like all the videos of the under the ocean are just like so peaceful and pleasant.
And the music.
to watch. Yeah.
I like the
The
music.
music
My
is
sister
good.
hates it so much because she's like so scared of sharks. So if there's ever like an
Ellery
underwater
is?
video,
Oh,
yeah,
really?
she hates it. I'm like, why?
Yeah.
There's no sharks. It's fine.
Okay, so yes, so Ruby said that is how they get their food, and they're getting little pieces of algae, plankton, bacteria. They are they're like a filter feeder, so it's going through. They're filtering out that food. Does anybody remember how they get the food? When they're filtering that water through, how do they get the food? Does anybody remember what it is that the food gets caught on? Essie?
The color cells.
Yes, so the collar cells, which are also called choanocytes, remember that root sites always means cells, so the official scientific name is a choanocyte, but they are collar cells, and it has like a sticky collar on the edge, and that is where the food sticks to that. And I'm not seeing that a sponge has a digestive system like we do. So how, when the collar cell catches that sticky food, how is that food going to get broken down and turned into usable nutrients? Eli?
Amoebocytes, move them around.
Okay, so, and the amoeba, so you've got your, kind of your rows of your collar cells, okay, and those are lining the big tunnel that the water's coming through. And then you have a layer of mesenchyme, which is like a jelly-like substance, and in that mesenchyme you have what Eli said, the amoebocytes, which are these cells that are taking care of the digestion, okay? And then those amoebocytes are going to release any waste out, and that's just gonna go right out the sponge, just like that water's pumping through. What else is in that mesenchyme besides the amoebocyte? Does anybody remember this? And we're gonna look at this under the microscope.
Can you repeat the question?
So inside that mesenchyme, okay, so you have the collar cells are kind of lining the inside of the sponge. And then you have the mesenchyme, which is like a jelly-like layer, and it has the amoebocytes in it that do the digestion, and then it has something else in the mesenchyme.
The ganglion?
No, but that's a good guess. Sounds close to that. And they come in different shapes, and they might make a sponge feel spiny or spiky.
The spicules?
Yes.
Stop! I was like,
So the spicules, and spicules can be different shapes, and so that's one of the slides that we're gonna be, we've got 2 slides of sponges that we're gonna look at under the microscope, so you can look at the spicules and can describe them, and so you might have like a hook shape, a barbed wire fence that's kind of with the pokies. It might just be kind of a spiky shape. And so you're going to be looking for the spicules. And sponges either have spicules or they have a spongin, okay? Sponges that have spicules are gonna be like a little pokier and harder, and the ones that have spongin are going to be softer, okay? And so, you guys, at some point you can come up here and touch these. I don't know. These to me don't feel spiky. I think of more of the, but this part over here does.
Maybe you should put them in water and see if they get soft.
Well, we could do that. We could do that. I did put this one under the microscope. I took a little sliver of it. One of the slides has got this one. And I think that I see some spicules in it, but you all can tell me what you think. Okay, but you know those, my mom used to get these, the loofah sponges, you know what I'm talking about, that people might use on their feet to kind of like scrub. They're kind of abrasive. Those would definitely have spicules to give you that kind of abrasiveness. All right. Okay. Tell me about sponge reproduction. So we talked about with the harvesting, they can regenerate, and so that's going to be an asexual reproduction, and what is that going to be called? Budding. Okay, so they can bud, just like we've seen other organisms before, where you kind of start forming cells, and this bud grows and grows, and then it becomes its own separate organism, finds its little spot on the ocean floor. So budding, and then also regeneration, growing parts that have been cut off. So when the harvesters are cutting off, they're gonna regrow. What do you think the sponges must have in order to regenerate? Think about the hydro video. Anybody know? This is not in the book. This is just like kind of making a connection.
They need stem cells.
Stem cells. Okay, so stem cells is what really allows that regeneration, and we're definitely gonna study several organisms, this semester that have a high percentage of stem cells. So the book didn't say anything about sponges having a lot of stem cells, but my guess is that sponges are probably not like the hydra, who 50% of the hydra cells are stem cells, which is why the hydra is always regenerating. But that sponges must have some stem cells. And stem cells are kind of like special cells that aren't a certain kind. They're undifferentiated, and they can differentiate. They can basically become any kind of cell they need to become. And so that's what gives the hydra that special ability. So my guess is that sponges, because they regenerate, have got a nice supply of stem cells. Okay, and so there's one other besides the budding and the regeneration, one other form of asexual reproduction for Porophora. And Ruby, you said something about this earlier. So I'm thinking gemule, but maybe not. You might. So
Maybe it's to have like the way that you pronounce it.
It does.
Oh, does it?
Oh, it does.
Gemule? Okay. Okay.
Bro, okay, whatever.
Like a gem.
They need to have this.
Okay, and so that's going to be, kind of remind me of like a cyst that an amoeba forms. So it's basically going to be covered, it's kind of like a little embryo covered in a hard spiky shell that's going to go find its spot, and when conditions are ideal, then it will grow into a new sponge. Okay, so those are going to be the asexual types of reproduction. Do sponges do sexual reproduction?
Yes.
And how do they do that?
They both, like, they could produce an egg or a sperm, and then it just releases into the water, and then it like finds another one.
Okay, yes, so the egg doesn't release into the water. Okay, the sperm do. The egg stays up in the sponge, okay?
And then doesn't like
I thought it was very unclear in the book, Ruby. I actually have to
Very
go
short.
back and
It was
like
like-
Google it and look at it.
It says these gametes are then released into the flow of water.
It did, and I went back and looked at it because I had written a note last time I taught it. But and the other thing that was confusing to me is that it made it look like, that, so basically most sponges can produce eggs and sperm, okay? But they typically produce sperm at a specific time of the year, when they're spawning. And they do release the sperm into the water, and they're going to go swim to another sponge. And the egg is actually inside the sponge, and those choanocytes are gonna help. They're going to, just like the way that they're filtering the water through, they're gonna help get that sperm to the egg, okay?
That's what jellyfish do too.
Yes, it's gonna be similar to that. Okay. And then when the, sperm gets to the egg, you're going to have a larvae that's gonna have some cilia attached. Remember how cilia help something move? Cilia are often attached to cells, and they're gonna help the cell move. But then that larvae with the little swimming cilia is gonna move till it finds a special place to attach, a suitable place to attach, and then it's gonna grow into a sponge. All right, what about predators for sponges?
No.
Sea turtles?
Sea turtles. Okay, there's really not a lot of predators, but sea turtles are a predator. Sea turtles will also eat jellyfish. Yes. Okay. All right, good. Okay. So what is our next phylum? Moving along.
Nadaria?
Daria.
Yes.
It's
I
don't know if the C is silent.
you what?
I don't know if the C is
Nadaria, yeah.
It is. It's silent.
Who needs
Yes.
to see?
And so that's going to include your hydra that we studied, your anemone that we've talked about some, and your jellyfish. And tell me about the two basic forms in this phylum, Ruby.
A polyp and Medusa.
Okay, and what's the polyp like?
Doesn't it like stay Cecil, but then the Medusa goes like out?
Yes.
And
what's
look,
it, yes. So the polyp is, yes, the polyp doesn't move. The Medusa does move. And what kind of symmetry do they both have? Anybody know that one?
Is it radio or?
Yeah.
Is it?
Radial.
Oh, radio.
Radial.
Oh, right.
Yeah,
Why'd you laugh at me? I got it right. He said radio.
No,
Radial.
I thought she was trying to say radical.
Okay, so
That radical symmetry.
they both have radial symmetry. And the polyp form is kind of like like a tubular shape, like the anemone, and it's gonna have tentacles on one end with a mouth kind of down in there at the top, and then a basal disc on the bottom. And then the medusa is going to be kind of a bell-shaped body with tentacles.
A
underneath.
jellyfish.
Yeah, like a jellyfish, yes. But a jellyfish is also a polyp at certain stages of its life. So, so you will see that in more detail tomorrow. All right, so I want everybody to go ahead and turn to page 338. And we're looking at the hydra. And one thing you'll notice this semester is that as we progress throughout the semester, the digestive system in these animals gets more and more complicated, more and more complex. So when we just did the sponges, the digestion was happening just on the cellular level, the amoebocytes. And now we're getting into the hydra, and the hydra actually has a mouth, okay, which is kind of our entry point for our digestive system. And because I am a dietitian, I do talk about digestive systems with all animals and compare it to ours. I like to talk about digestive systems. So that'll be a theme throughout the semester. But so the hydra has a mouth, and that digestion, okay, so when that food goes in to that middle area, that is called the mesoglea, that middle area. I'm sorry, no it's not. It's the gut of the hydra. The mesoglea is in between the cells. But that is just kind of like an open space with digestive enzymes in there that are released from that inner layer of cells, the gastrocytes, the gastrodermis layer. So you've got two layers of cells on the outside of that gut, and the inner cells are the gastrodermis, and think gastrointestinal. And so those are the cells that are going to release the digestive enzymes into that gut area where the digestion is going to happen, where the food's going to come in. They're going to sweep the food in with the tentacles. It's going to go in through the mouth. It's going to be in that gut area, and then those cells on the inner membrane are going to release those enzymes that can break down the food into the gut, and then the waste will actually go through the cells through osmosis and diffusion, and then just diffuse out of the body. The waste doesn't go back up through the mouth, it's going out through the cells. Does that make sense? Okay, so this is a very simplified digestive system. So we've got the two layers of cells. The outside layer is the epidermis. Epidermis always, so when we talk about our outside of our skin, that's our epidermis. So epidermis, epi is always on the edge, on the outside. So those are the outer cells. The inner cells are the gastrodermis, and then in between those two layers of cells is the mesoglea, which is just a jelly-like substance between the two layers. And then that center part is the gut. Those little lines are supposed to represent food coming into the gut. Okay, so what about the bud? What is that?
That is how they asexually reproduce.
Yes, so that's what the bud is. So we're going to be looking at some hydra under the microscopes today. They're going to have buds on them. So you're going to be looking for the bud, and you're going to be drawing the bud, okay? They're going to have tentacles. There's one of the hydras that actually has a broken off tentacle on our slide, and you can see the tentacle near it. But good thing for that tentacle, that broken off tentacle, what do you think is going to happen to that? This would be great to put in your conclusion. What's going to happen?
Regenerates.
Well, the hydra itself would grow a new tentacle, but what do you think would happen to that tentacle? Nancy, think. What? No, what else? What's gonna happen to a tentacle that's broken off? What's it gonna turn into eventually? Because of the amazing power of
A
stem cells.
new hydra.
Yes, yes. So that would be a great thing to talk about, in there. Okay, so you see that basal disc on the bottom? All of the, that's not just, that's all polyps are gonna have that basal disc on the bottom. That's gonna help it stick to the bottom, help it root itself wherever it finds its home. And then you have the testes bump and the ovary bump. Most hydra are gonna have one or the other. They're not necessarily gonna have both, okay? So you're gonna look on that side and you're gonna look for a testes bump or an ovary bump. Are you gonna be able to tell which one it is?
Yeah, maybe no.
I don't think so. Yeah.
If one is higher up, is that what they're saying?
No, I don't think so. I don't think that our microscope's gonna be, you can tell in this picture, the ovary doesn't have as many, has larger cells in it, and they're bigger, and the testes has a bunch of smaller sperm cells. I don't think our microscope's gonna be powerful enough to be able to see. You can try, but I don't think so. And then tell me about the tentacles and what is on those tentacles. Nancy?
They're called nematocytes. Nice. Yeah, sorry. I do have
Cysts.
to, yeah, I have a list.
I say
right now. This
nematocyst.
is one.
Nematocyst, is that easier to say?
Yeah.
Nematocyst.
Nematocysts, yeah. And they're basically like these toxic, like, if somebody brushed up against it, these toxic, I don't know how to describe them, like, something that can sting somebody.
Right. They could be injected into
Yeah.
prey.
And it's released, and then,
Yes, so that when you get stung by a jellyfish, you got pricked by the nematocyst. That's what it was that injected that jellyfish sting into you. That's exactly what it is. Okay. Let's see here. All right, so we're gonna look at that in a minute. The nematocyst, does anybody remember how the nematocysts on the hydra are different than the nematocysts on the anemone, Ruby?
On the hydra, it's pressure sensitive, so like if someone touches it, but on the anemone, it's based on like the, not chemical, but just like, I guess chemical and like how it reacts to it. It's like certain things it's gonna.
So with the pressure sensitivity, it might not even be that something touches it, but it feels the water pressure, like there's some kind of other animal in the vicinity, okay? That's gonna trigger it. With the chemical sensitivity, it's gonna sense some kind of chemical in the water, okay? But which different organisms kind of give off different chemical signals. But because that clownfish, does anybody remember what that clownfish is covered in that makes it not give off the chemical?
Mucus.
The mucus. So the mucus stops that chemical signal, and that's why the anemone does not shoot off their nematocytes for the clownfish. Okay, now let's go ahead and talk about the corals, which are also known as the cnidarians. And what shape are the corals down in the, remember the coral itself, what we think of as the coral reef, that's where it lives, down in there, but what is it actually that's in there that's hiding down in there? Yes,
It's
Eli?
little cup shapes.
Yeah, but what is the official shape of that, remember?
Is it radial?
That's the kind of symmetry it has, but remember we had two shapes for
Oh, polyp.
polyp, okay? So it's going to be a polyp shape down in that coral, and it is, that the cup itself is contracted when it's not feeding. All right, so coral reefs. One of the things I want to tell you that's really important about coral reefs that wasn't in the chapter is that coral reefs help protect us against storm surges. And so I guess because they just kind of help, it's like a buffer. It helps prevent with hurricanes and all that kind of stuff. It helps prevent as high of a surge. So it's very important that we protect our coral reefs, and there's entire ecosystems in these reefs. What are things that can cause damage to coral reefs?
I guess like having a boat that's too shallow.
Yes, so like man-made, somebody hitting it. Some people want to go and like cut coral out or do damage to it. That's something. Pollution, different chemicals and stuff interacting with it. The oceans getting warmer can do damage. The fluctuations in ocean temperature. That can cause damage, but also if you go somewhere where there's coral reefs and you do like a snorkeling trip or scuba diving, it's really important that you don't wear regular sunscreen, because the regular sunscreen can it all comes off, and when you have a coral reef area and it's a tourist area, you've got lots of people coming through there all the time that have sunscreen that's going to wash off on them, and that sunscreen can affect the organisms in there. So you always want to, if you do that, you want to buy this special reef-friendly sunscreen, which is kind of sticky and not great to wear, and it's expensive, but it is important to protect the coral reefs. Okay. Anybody have any questions about the corals, the Nidaria or the coral reefs? Okay. Has anybody ever been snorkeling and seen some?
I did in Hawaii once. It was so much
Okay,
fun.
Hawaii.
Yeah, I actually got baptized in Hawaii. It was a great trip.
Wow.
But yeah, it was really cool to see. There were like the what are they called that are like, they're spiky and they're brown, but they just like sit on like the coral and like stuff like that.
Urchins, sea
Urchins.
urchins.
There were a
Yeah.
lot of those.
A
I
lot
was
of
pretty
urchins.
scared.
Yeah, that
But
would be
also, my dad and my brother saw a sea turtle like right next to them. I didn't see it. I was mad, but like while we were snorkeling, they saw that. But it was really cool.
How old are you?
9.
If you fly to an area that has coral reef around it, it's really neat on the airplane. As you get lower, when you're overhead, you can see the reefs like in the water, like where they're going. And you can see that view of them really helps you see how it would protect the land. So, all right, cool. All right, so now we're going on to the jellyfish, and the big thing that I want you to understand about the jellyfish, and again, I'm going to send you another video that I want you to see with the jellyfish, is the jellyfish life cycle. So what we think of as jellyfish is just really a small part of their life cycle. And so we've got the boy jellyfish. I'm on page 341. If everybody could be on page 341. At the top, we've got the boy jellyfish, and he's releasing his sperm into the water. The egg is inside the girl jellyfish. That sperm's going to travel up to the girl jellyfish. A zygote's going to be formed. It's going to become a planula, which is basically like a little baby jellyfish embryo. It's going to have cilia attached so it can swim. It's going to attach itself to the bottom. It's going to start growing and start forming into a polyp. And that polyp can bud and form more polyps asexually. And this video that I'm going to show you has some really cool pictures of that, which I feel like you kind of saw this somewhat. You saw the polyps just everywhere in that video that you did watch. The polyp is going to grow and develop, and then it's going to form these discs one by one. Each one of those discs becomes the new Medusa. And this cool thing about this video I'm going to send is you actually see the discs one after one just like getting released from the polyp. And so one planula, okay, can end up forming lots and lots of medusas. Because one planula, it starts with a polyp, but then that polyp is gonna asexually bud and reproduce. And then each of those new polyps then has multiple medusas that get released, okay? But jellyfish do have predators, and so typically the predator, even though you're gonna have lots and lots released, the predators are going to keep the jellyfish in check. Occasionally, ecosystems will get out of balance with jellyfish, maybe because there's a lack of the predators. I know that when I was growing up, there were probably 2 different times that we went to the beach, and it seems like it was always a little closer to late July or August. It wasn't earlier in the summer where they were just having jellyfish explosions, and you would just see them washed all over.
Yeah, when
Have
I
you
go,
seen that?
yeah, when I lived in Charleston, like every time we go to the beach, there are so many dead jellyfish.
Did you feel like it was kind of at a certain time of year that it was worse?
Yeah, I think so.
Yeah.
I
Okay.
feel like it was like one month we would go and they would all be dead.
Yes. I think that happens. And so, anyway, but that when jellyfish are like that, I mean, it's really scary to be in the water like that when there's just tons and tons of jellyfish. That is not a pleasant thing. Okay, so on your own on 11.7. A lot of animals need some sort of respiratory and excretion system. Why don't cnidarians need these?
I don't know, because they have such thin epithelial layers that gases and waste products can be directly exchanged with the surroundings.
Yes, it's osmosis and diffusion. Okay, it's just going back and forth between the cells. Good. Okay. If a jellyfish reproduces asexually, is it in the polyp form or the medusa form? Polyp, asexual. Asexual. Yes, sexual when it's in the medusa, Nancy. Asexual when it's in the polyp. Okay, all right. So, I'm going to kind of go over these labs. And so everybody can flip to page 334. And what I have, the 2 microscopes on the other end have sponges, okay? The one on the very end that's up against the wall has a very thin slice of this, okay? And then the one that's behind Eli is a prepared slide from the box. And it is only the spicules from a sponge, so they removed the other material and it's only the spicules. My slide that I made, I just cut a very thin piece of my sponge and did a wet mount. Now you'll see kind of a dark line, that's just the water because I did drop some water on it. And so hopefully you can look at both of those and draw both of them. I think you'll probably get the best views on them on 10x. Okay. And remember to make sure you read the instructions because the instructions are going to tell you what you're looking for. But they're definitely looking for you're looking for the spicules. You're describing the shapes of the spicules and drawing them. All right. For that one, and then you can also come up here and look at these if you want to. All right, then. So you should have at least a drawing of, at least one drawing. You might want to draw both of those, the piece of the natural sponge and the prepared slide. And then you should note, maybe even label on your drawings what the spicules are in your conclusion. You're going to talk about what shape spicules you saw. Okay, is that making sense?
So draw one of those, or
No, just
Okay,
the
you
slides.
put it there,
Yes,
so-
this is just kind of here for you to look at. You do not have to draw these, okay? Yes, that's not part of the lab. Just the 2 slides are. Okay. Then you're going to flip to 339. This is our other lab. So this is going to be written up as 2 labs. So it's going to be 2 entries in your table of contents, 2 separate labs. And you're going to have the microscope by the piano and then the one next to it both have hydra slides. Okay, the one by the piano is the one that has the broken tentacle and it's dyed differently than the other one, but they both look good. And so you're going to have 3 drawings for this lab. I always prefer it color, pencil, just so it's just kind of easier to see. And Eli, I have big expectations of you now that I know you're like art.
Well, I
So.
don't have a colored pencil.
Well, colored pencils are on my class list, so they're actually supposed to be in your bag.
I mean, I just,
Okay. But you can also do your drawings after the fact. But, you know, just maybe do it on scratch paper today and then put it in your lab notebook neatly. But anyway, those 2 are gonna be the Hydra. The Hydra, I'm looking at, you're gonna have 3 drawings on that Hydra lab. One is going to be, and you'll see this in the directions, okay? One is going to be you're drawing the entire Hydra, and that's gonna be, if you mark in your book, by procedure number 3. You're drawing the Hydra and you're labeling tentacles, mouth, bud, if one exists, the basal disc, the nematocyst, testes or ovary, if one is present. So you're drawing the Hydra as a whole, that's probably gonna be under 40x. Both of those are set up on 40x right now with the Hydra as a whole, okay? That's gonna be your first drawing. Your second drawing is going to be on number 6, and you're going to focus in on the tentacle, and you're going to draw that. And you're going to make sure you can identify the nematocyst, and you're gonna make sure you label that on there. So you can mark number 6. And then number 10 is your last drawing, and it says, draw a portion of the wall of the body. So you might focus in on that body wall where you have those 2 layers of cells, the gastrodermis and the epidermis with the mesoglea in between, and you draw those 2 layers. So that is the other one. And so make sure on your drawings that you always put out to the side what side of your microscope was on. I think on the Hydra, you might feel differently. I got the Hydra as a whole under 40x, under the lowest setting, 40x, and the close-up shots for the tentacles and the wall of the body under 10x, and for the spicules, I got the best views under 10x. So if I drew them, that's probably the magnifications I would use, but you're welcome to mess around with them. And so you're gonna kind of just do both labs at once, but you're gonna make sure you write them up separately. Does everybody understand? So the Hydra, you've got 3 drawings, and what you may want to do is take a picture, do kind of a sketch, and then you go back with your textbook and make sure you get it all correctly labeled. You might want to sit there and know in your mind what each thing is, and then you do your drawings. Is everybody with me? Okay. All right. Did everybody pick up maybe on some ideas and be thinking about ideas for the conclusion? Okay. All right. And make sure you take the time to read the labs. All right. Get to it. And Anna, if you could get photos, so I want you to get whatever Hydra you look at, I want you to get 3 photos. The whole body in on the tentacle and in on the body wall, and then one or 2 of the spicules.
I can probably just get a picture of all 4 of the slides.
Well, you only need one on the Hydra. You just need to get on one Hydra, but you need to get 3 images because you're supposed to focus in on 3 different things. So Hannah, listen, so the Hydra as a whole, which is on 40x, because you're looking at the whole body, then zoomed in on a tentacle, and zoomed in on the side of the body wall, because those are the 3 you're in front of the camera. Actually, I can go ahead and turn this off.
Is this one the sponge, right?
Yes, that is the